So topic 10, we are back to talking about respiration. And if you remember last day, we had talked about the first three components of this. We talked about glycolysis, pyruvate oxidation, and the citric acid cycle. And at that point, we had made 2 ATP in glycolysis, and we made 2 ATP in the citric acid cycle. And that was it. Only 4 ATP so far. But what we also have made was a bunch of NADH and a little bit of FADH too. And so this is going to be used in the electron transport chain. And this whole process here is called oxidative phosphorylation. So that's what we want to talk about today. So before we get into oxidative phosphorylation, I want to talk a little bit more about the mitochondria. I told you I was going to make you a diagram and label it for you. So I showed you this diagram here last day. And so the citric acid cycle takes place on the innermost component, which is the matrix. So I'll show you some pictures of some mitochondria, and then I'm going to draw one for you. Here's another mitochondrion, and you can see this is the kind of from that series. I think I showed you a few of these before with that sort of half cartoon, half electron micrograph. And you can see all the internal structures. So those internal membranes are going to be really important. And there's some more mitochondrial cross sections, and there's lots of nice pictures of mitochondria. So there's a mitochondria I drew for you. Usually I draw one on the board, but I found this nice model. Often I draw it and it kind of looks like this. So you can see that my drawing is not quite as nice, but if you want to draw something like that, that's great. And so what I want to do is label some components and kind of talk about what's going on in each of the parts. So the first part is the outermost component, and this is, I don't know why I used an O or an I meant to use O, and this is the outer membrane. So the outer membrane is relatively porous and mostly structural. So maybe I'll put a component here, or a note. Mostly structural, meaning it doesn't have a lot of biological function other than holding the mitochondria, kind of just containing everything. So the in between there is something called the, let me just do a couple of labels. The other membrane is called the inner membrane. And in between is called the intermembrane space. So intermembrane space. Okay, I'll come back to that one in a moment. I just left some space, but the inner membrane. So the inner membrane, this is the location of the electron transport chain. So ETC is electron transport chain. And this is also the location of ATP synthase. So this is good. We're going to make a bunch of ATP there. So that last part here, the intermembrane space, this is going to be the location of something called the proton motive force. Location of proton motive force. So another way to describe that is to say this is where the protons are pumped. So this is where protons, so H+, are pumped. So we'll be talking about in a minute about what that means, because we are going to be talking about the electron transport chain and oxidative phosphorylation as a whole. So what does that leave? That leaves the last part, which is the matrix, which is the part we've talked about the most. So that's the innermost component, so matrix. And so we've already talked about this being the location of the DNA. So location of, we had DNA and we had ribosomes. This was also the location of the citric acid cycle. So citric acid cycle. And one more thing I'm going to throw down here as a note. We're not really going to talk about it in this course, but just sort of for your information, is that this is the location of something called beta oxidation. So that's a beta oxidation. And so what do I mean by beta oxidation? That is where we consume and oxidize our fatty acids. So this is fatty acid breakdown. And I'm mentioning this because I mentioned before that, I actually mentioned this before when we were talking about peroxisomes. I had mentioned that peroxisomes is where we have weird fatty acids get broken down. So if they're branched or extra long, they go to the peroxisome first before they go to the mitochondria. Normal fatty acids go to the mitochondria and they get broken down, and they actually get broken down into acetyl-CoA. So acetyl-CoA is, of course, the feeder molecule that goes into the citric acid cycle. So I'm just going to underline these things as the components, just to make my diagram a little tidier. There we go. And so that's kind of our introduction to mitochondria. They do have other biological functions, by the way. Mitochondria are involved in something called apoptosis, which is cell death, and there's a few other things that happen in the mitochondria, but their main thing is a place to make ATP, where things are going to get broken down, and so we're going to make those electron carriers and ultimately make ATP out of them. So let's move on. This is a short video I'm going to play for you. I think it's only about 1 minute long, and it's a little has some corny music, but what I'm going to do is play it for you. It's kind of going to describe what's going on with this whole process, and then I'll explain it again using the images from the textbook. So a couple of things I want to point out before talking about oxidative phosphorylation is that there's actually two parts to this word. So there's the oxidative part, and this is actually the electron transport chain. Okay, this the electron transport chain is actually only part of oxidative phosphorylation. And the phosphorylation part is the ATP synthesis. So notice down here, I call it chemiosmosis. That's another name for ATP synthesis, and we'll see what that means in a moment. So let me play this video for you, and then we'll go over it in a little bit more detail. Of course, in order to play the video has to work. So that's wonderful. Just give me a moment here. I'll see if I have a different version of the video. I think, let me see here. My computer is thinking. Okay, so
electron transport phosphorylation, the final stage of aerobic respiration,
takes, but the other version doesn't have sound. Okay, so ignore the video. I apologize. So let's take a look at the image from the textbook then. So if you take a look, let's just look at some parts of this for starters. So first of all, we have our NADH. It's way down here. Okay, so remember that was a product of glycolysis, a product of pyruvate oxidation, and it was also a product of the citric acid cycle. We also have some FADH2 as well. And so those are the products. Remember what they have done is they've taken electrons in bonds to connect to the hydrogens. And so those electrons they have energy in them. And so now what we're going to do is we're going to oxidize the NADH and oxidize the FADH2, and we're going to harvest the energy from it. So when we oxidize these things, notice NADH turns into NAD+, so that's going to get recycled. FADH2 is going to get recycled back to FAD. So the next question is, what happens to those electrons, and what is going on in terms of harvesting the energy? So take a look here. This is the electron transport chain, and it's got a bunch of these massive protein complexes. So they all have numbers. We've got 1, 2, 3, 4. Sometimes we call this one 5. We'll talk about that one in a minute. There's some other things in the middle. We've got this Q that stands for ubiquinone and cytochrome C. So there's a whole bunch of components to it. I don't need you to know the names of all the components, not in this class. But what you can see is going on is that the electrons from that bond in NADH are transferred to the first protein, and then they're transferred to the next one, and the next one, and the next one. So if you take a look at this one, it goes from complex 1 to Q, to complex 3 to cytochrome C, complex 4, and eventually, during this transfer, those electrons end up on that final product, and that final product is oxygen. And so you can see what happens. We get 2 electrons, and we get a couple of protons, and eventually we get water. So water, or oxygen, is gaining electrons, so that means it gets reduced. And so oxygen is our final electron acceptor, or our terminal electron acceptor. So that's where the electrons go. In the meantime, when those electrons are moving down this chain of proteins and getting transferred, something else very important is happening. And so protons, they're not shown down here, but I can draw some. They are going from the inside of the mitochondria. So they're going from the matrix. It's not labeled down here, but I can label it. This is the matrix. And up here is the intermembrane space. Intermembrane space, there we go. And so those protons are being transferred or pumped from the inside in the matrix, there should be an X down there, to the intermembrane space. So up here, now we have all these protons. Some are getting transferred or pumped by complex 1, some by complex 3, some by complex 4. Complex 2 doesn't pump any protons. Not sure why that is, but it doesn't really matter. What does matter is we have a whole bunch of protons that are ending up in the intermembrane space, and what we're getting here is something that we call a proton motive force. You can also call it a proton gradient. Meaning that we have a high concentration of these hydrogen ions in the intermembrane space and a low concentration way down there in the matrix. So that is the electron transport chain in a nutshell. It's basically taken the electrons from NADH and a few more from FADH2. You can see FADH2 enters the pathway not at the beginning, but at a second place, so it's complex 2. It's lower energy, so it can't enter in door 1. It has to go in door 2. And that's the electron transport chain. It moves those electrons through the chain, and the electrons end up on oxygen. We make water. And during that process, we make this proton motive force. And so this is an electrochemical gradient. And what that means is we have a concentration difference over a membrane. And you probably know that that concentrations don't like to be different. Ions like to move from a high concentration to a low concentration. And so that's the second part of this. The first part is the electron transport chain. The second part is chemiosmosis. I prefer to call it ATP synthesis because that describes better what's going on. But you can see these protons here. They actually travel through this enzyme called ATP synthase. And as they travel through, what they do is they affect the active site on this enzyme. And I'll show you a little animation here in a moment. But what actually happens, believe it or not, is as the protons move through this, the bottom part, not the bottom part, this part of the enzyme actually rotates. And it's kind of like a little machine. And as it rotates, the active site kind of opens and closes. So I kind of think of it as a little bit like one of those doors. I think we have one at the airport that rotates, and so it opens, and you gotta, you sort of gotta get in there with your suitcase before it closes. And that's what's happening, right? As the door, in this case, the active site opens, ADP and inorganic phosphate get in there. And as it closes, it pushes them together and it forms a bond, and that bond is making ATP. So what we have is we have an electrochemical gradient that gets converted into movement, and that movement, the kinetic energy in that movement is used to form chemical bonds. It's really a very cool process in terms of chemistry, all the things that are going on here. I think it's just amazing. So I'll show you some pictures of this, but this altogether is a process we call oxidative phosphorylation. So I kind of like this because it's got two words. The oxidative part includes oxygen, and the phosphorylation part is where we're adding phosphate to ADP to make ATP. So two parts, two words. Hopefully that's easy to remember. Before I move on and show you some pictures of ATP synthase, I just want to point out a couple of things. So what if we don't have oxygen? So the answer is we die. That's the short answer, at least in the long term. In the short term, we might be okay. You can probably hold your breath for, you know, short amount of time and do just okay. But if we don't have oxygen, this actually shuts down all the pathways except for glycolysis, because all the pathways are kind of like this big, you know, assembly line, right? And this is the last part of that assembly line. So if this part of this entire sequence stops, then everything before then cannot move forward. So this shuts down everything except for glycolysis, and I'll come back and talk about that in a moment. There are some poisons that can stop this, by the way. So you may have heard of this little poison called cyanide. So cyanide will actually stop the electron transport chain right there at cytochrome C, and so that's how it kills people. It's kind of like, I guess, suffocating to death. We can't get our electrons to oxygen, and our cells just, you know, they shut down and die relatively quickly. I don't know if cyanide actually makes you foam at the mouth, like they show in the movies when there's somebody taking a cyanide tablet and if it kills you within seconds, but it is a pretty potent kind of poison. So back to ATP synthase, I told you I had some pictures to show you. There's another kind of nicer picture, and you can see the protons are going in. You've got this rotational movement here, and like I said, that part sort of rotates, and it causes this the purple stator thing here. The stator kind of it flexes back and forth and opens and closes the catalytic sites on this enzyme. It's really quite cool. And I think I have a video here I'll show you in a moment. You can see there's sort of a bit of a formula. It says it takes 2 to 3 protons to make ATP, and I'll give you a more precise formula in terms of the reason why it's 2 to 3. Is that, you know, depending on the textbook, some people are talking about, you know, you have the ATP and it has to be transported eventually out of the mitochondria, and there's a cost to that. So some people add the cost in and some people don't. But here's a formula for you, is that every NADH that goes through the system makes 2.5 ATP. And every FADH2 makes 1.5 ATP. So I'll come back to that number in a minute. I just want to show you a couple of things that are going on with this ATP synthase. So this is a little video. I'll play this for you. Of course, that was not working too. This is just, I don't know what's going on here with my videos. This one shows a little bit of motion in that, and I think I showed you this before, this image, and just as an example of an enzyme that has quite a number of different subunits, at least 15 subunits. So 2 things to say about this. First of all, is that we already talked about something that uses the proton motive force, and something that causes rotation. So you're probably thinking, wait a second, what else have we been talking about in Biology 107 that rotates? And the proton motive force, so it powers 2 things, at least 2 things anyway. ATP synthesis, we just talked about now, ATP synthesis. And the other thing that rotates is your prokaryotic flagella. So remember, our prokaryotic flagella rotate, and when I drew those pictures for you a few lectures ago, I was showing you that you know, the eukaryotic flagella is big, it's covered in membrane, it uses ATP. The prokaryotic flagella, so in bacteria like E. coli, it doesn't use ATP. It actually uses the proton motive force, and it rotates, and it's kind of a little bit like ATP synthase in that way. The other note is just a footnote here as well, is that this whole thing can be run in reverse. So what does that mean? It means I can break down ATP and I can move protons. And so, for example, your stomach has a lot of H+ in it because your stomach is acidic. And so we can make organelles like lysosomes, or we can make organs acidic by consuming ATP. So just another little footnote for you. So I think, let's see if this video works here. It's not loading up. There we go, now it's loading up. My screen looks really weird, but I'm gonna play it anyway and see what happens.
ATP synthase is a molecular machine that works like a turbine.
On this. So let me just see if I can find the actual file. And I'm not sure what's going on with my computer sound today. So just give me a moment here. I'm gonna go find that file. Okay, I think, which one is it now? It's not that one, not that one. So hold on a second, I'm just trying to figure out if I can find that file. Let me just go back to the PowerPoint here and see if that works again now. Oh, somebody said they were able to hear it. Okay, well, I'll play it for you. I don't know why I can't hear it, but let's go.
ATP synthase is a molecular machine that works like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP. The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP. The F1 ATPase portion of ATP synthase has been crystallized. Its molecular structure shows that the position of the central shaft influences the conformation and arrangement of the surrounding subunits. It is these changes that drive the synthesis of ATP from ADP. In this animated model,
So I'm just going to stop there because that's kind of what I wanted to show you. You can see how the rotation leads to the ATP synthesis. If you want to watch the whole video, just download the PowerPoint, and the link to the video is there. You can check it out. So here is kind of the summary of this, and what I want to do for a moment is actually just going to go to the whiteboard and kind of tally up what is going on here, but in terms of all of these processes. And you can see they're giving a number here, about 30 or 32 ATP. So let me just change to the whiteboard, and what I want to do is just kind of go through these and give a recap of what's going on with the ATP production. So we had basically three processes before we got to the oxidative phosphorylation. So we had glycolysis, and we had pyruvate oxidation, and we had the citric acid cycle. So a little bit of stoichiometry for you. So let's just try to remember what everything added up to be. So remember, in glycolysis, we made 2 ATP. So what I'm going to do is basically count up all of our ATP, by the way. So 2 ATP, there we go, 2 ATP. Okay, pyruvate oxidation, there were no ATP being made. And the citric acid cycle, we made 2 GTP. Remember, GTP is exactly equivalent to ATP. So there was our 4, and so that wasn't very much, right? And but what we want to do now is consider all of the other things that we've produced. So remember, in glycolysis, we made 2 NADH. And pyruvate oxidation, we also made 2 NADH. And the citric acid cycle, if you go back and count, we made 3 NADH, but I'm multiplying it by 2 because of 2 pyruvate that ultimately are put into the system. So that becomes 6 NADH. And in citric acid cycle, we also made 2 FADH2. Okay, so what does that mean we're getting here? Going back to that formula, remember the formula was that we had for every NADH, it makes 2.5, so I'm going to multiply that by 2.5. Multiply all of my NADHs by 2.5, and my FADH I'm going to multiply by 1.5. So how many ATP does that make? So 2 times 2.5 is, of course, 5 ATP. 5 here. And the citric acid cycle, I have 6 times 2.5. I have to think about that for a moment. That is 15 ATP, so that's pretty good, considering we only had 4 before. And 2 times 1.5, of course, is 3 ATP. So that is all my energy production, and just draw a line here. And what is the total? Add all those up, and you should get 32 ATP. So depending which textbook and all those kind of things you'll look at, sometimes you get different numbers. And there's different reasons for that. I think I mentioned that in order to get the ATP out of the mitochondria, there's a cost to that to transport things across membranes if you want to use them for other cellular components. And in fact, that's why I have 2.5, because the reality is every NADH actually makes 3 ATP, but the net is 2.5 once you get that ATP out of the mitochondria. Sometimes different tissues give different numbers, like muscle cells I think are a little bit more efficient in making ATP, so the number is closer to 34, 36 or something like that. But this gives you an idea of where all the ATP comes from. Most of the ATP is coming from those electron carriers that are getting oxidized at the electron transport chain to make that proton motive force, which ultimately makes the ATP. So going back to this, you can see this is giving some numbers, and you can see it says about 26 or 28, and the textbook, at least this version, seems to agree with me and is giving a number of 30 or 32, again, depending on exactly how you count things. So I don't care about the exact numbers, but I'm just trying to, give you an idea of where all that ATP is coming from. And in fact, I think last semester on the midterm, I think I had a question asking exactly that. I didn't ask for exact numbers, but I said, where does most of the ATP come from? And one of those types of questions, I think it was a short answer question, and people were giving me lots of weird answers. So I don't know whether I worded the question poorly or not. I can't remember. I just remember lots of weird answers, something that I thought was obvious. So this says, what if there is no oxygen? Then, as I mentioned, all of these things get shut down, and we can only make 2 ATP, and that's not very much. So if you are a human being or a large animal, that is not enough ATP to survive for very long. We can survive in the short term without oxygen, but after a few minutes, it leads to death for us, unfortunately. So I want to talk about some of the physiological consequences of not having oxygen for a minute or two, and this is actually related to this week's lab. So this week's lab, we were talking about fermentation in yeast. So I'll get to the yeast model in a second. Let's just talk about mammals. So if you take a look, just take a look at the top of this slide here. We've got glucose, and the glucose here is getting turned into pyruvate. And in that process, we are making a couple of ATP. So we can do glycolysis without oxygen. And if I asked you to let's say do a bunch of push-ups, right? Just imagine you're doing your push-ups, and some of you I'm sure can do 100 of them in one sitting, but probably most of us after maybe 10 of them or less or a few more, you're going to get a burning sensation in your arms. And so what happens when you do vigorous exercise is that in those muscles and tissues, you have oxygen. It's there already, and it gets used up and depleted relatively quickly. And then eventually, the body just keeps doing glycolysis because, hey, you're telling it to keep moving. It needs ATP. So what it does is, in order to do glycolysis, you can see the other components of glycolysis is it's going to take NAD plus and make NADH. Now, normally that NADH is going to go onto the electron transport chain, but it can't because you have no oxygen. So instead what we do is we recycle the NADH, and you can see what it's doing here is it's making lactate or lactic acid. And so lactic acid is that kind of that burning feeling that you have in your muscles in the short term anyway. Eventually that gets broken down the liver and all that, and assuming you breathe again and get more oxygen, or maybe it stops you from exercising vigorously because your body doesn't like to feel that way. But this is part of the process. So you hear a lot of athletes talk about lactic acid and lactic acid buildup, particularly if you've done a lot of vigorous exercise for a while, and it can be really discomforting, but it's a natural part of our process. So what about those yeast? So I want to show you this video here that's talking about what goes on in yeast. By the way, this is what you came to lecture for today, is this video. This is a fantastic classic video. It's from an old, I think it's a BBC documentary, and I think it's called something like Animals Are Beautiful People. So I'm just going to play this for you, and we're going to watch and see what happens. It's a very, very good nature video. So I'm going to stop it there. You can always look this one up. You just need to look up drunk animals. You'll find it pretty quickly, but hopefully you get the idea there. This is usually what people mean when they talk about fermentation, is they're talking about alcohol fermentation, because of course people are familiar with the products of alcohol fermentation, the products being alcohol and carbon dioxide. So maybe you use yeast at home, maybe you're a home brewer or make your moonshine or something like that. But a lot of people use yeast at home for making bread products. You make pizza or homemade bread, and the carbon dioxide that's released will actually make the bread rise. And if you don't use it, I think I made pizza one time and forgot to add the yeast. It was edible, but not very tasty. In those cases, the ethanol it evaporates when you cook it, and it's not in high concentration anyway. But in the case of wine and beer products, of course, that's the desired product, and that's what happened to those animals there. So this is the pathway, and if you did yesterday's lab, hopefully you're relatively familiar with it now. You can see the first part is exactly what we saw before, glucose being converted to pyruvate, and in the yeast or some other microbes, rather than making lactic acid, they have a different biochemical pathway. So you can see in this biochemical pathway, it first makes something called acetaldehyde. And acetaldehyde is unfortunately something that is found in some alcoholic products. So if you've ever had a headache from drinking too much red wine, sometimes that's because the yeast in the wine making process haven't fully broken down the ethanol, and the acetaldehyde can give that headache. So don't drink too much red wine. And it eventually gets broken down to ethanol, which is, of course, the product people want. And you can see there's carbon dioxide in there as well. So that's what fermentation is, right? It's basically a partial breakdown of pyruvate. And the whole idea is to recycle the NADH and remake NAD+ so that we can continue with glycolysis. Now, in a human, that's a short-term solution to a problem of not having oxygen. In a microbe, they don't have these massive bodies and they don't need as much energy. So a lot of them can survive just on glycolysis alone. So this is just showing that basically, all organisms, we take glucose and we convert to pyruvate. And glycolysis is found in all organisms, by the way, at least in part. And like I said, it's kind of a universal pathway. And if we have oxygen and the mitochondrium, then we can do aerobic respiration. So we convert that into acetyl-CoA, and then that goes into the citric acid cycle. And then eventually we have our oxidative phosphorylation. You can see it's happening there at the inner membranes, and we're going to make a whole load of ATP over there. If you don't have oxygen, you can do fermentation, and you can make lactic acid, you can make ethanol, you can make a few other products too, just depending on who you are and what organism you are. So pyruvate is kind of like a branching point. And there it is. So just a couple other notes about respiration, and then we're going to finish up here. Prokaryotes, meaning mostly bacteria is what we're talking about, they have, there's a lot of different types out there, probably 4 million different types. And so they have a huge amount of diversity in some of the things they can do. So one thing that they have a huge amount of diversity in is that this thing called the terminal electron acceptor. So we are in group A, we are obligate aerobes, which means we require oxygen for respiration. And remember oxygen, remember we had that formula, we had the oxygen, we had our protons. I'm going to balance this equation for you. So if you want to balance it fully, you need a half a mole of oxygen and 2 electrons, and then we get water, right? So that's aerobic respiration. There's other organisms that are obligate anaerobes. So they don't use oxygen. So some of them use fermentation. There's a few organisms out there. You can see this one, this sort of subcategory called anaerobic respiration. So in that case, they might use something else like sulfur. So in the case of where they use sulfur, it's almost the exact same formula, except for we're going to substitute sulfur with oxygen. And what do we get? Not H2O, we get H2S. So that's how we get H2S, which is, believe it or not, a toxic gas. And they talk about a lot at some of the sites, the mining sites, because in some areas it just gets released. It's made by a biological process. So if you ever see that term, terminal electron acceptor, that just basically means you use oxygen or not. And it's talking about these different processes. So another footnote on this is that there's many different microbes out there using all sorts of different processes, and they can make us useful products. So we already talked about the ethanol here, and that's produced by yeast, of course, and some other microorganisms. But take a look at all these other products. We have lactic acid. Where do we find that? That's actually found in various yogurts. There's ethanol again. Propanoic acid, that's found in different food products, different yogurts and things like that. And acetic acid is vinegar, of course. We've got butanol, isopropyl alcohol, that's a rubbing alcohol. Acetone is nail polish remover. So a lot of these things, these fermentation products are basically products that humans use for a lot of different things, food and otherwise. So one other footnote to think about, something we talked about already, is that to regulate this whole thing, we have an enzyme up here called Phosphofructokinase. And I told you before that this enzyme is regulated by ADP and PEP. Believe it or not, it's actually also regulated by citrate from the citric acid cycle. And it's also regulated by ATP, which comes from downstream. So this is the big thing to know, is that this is a key regulatory enzyme, not just for glycolysis, but for this entire pathway. The last footnote is that these processes are not just used to break down glucose. So think about it, right? We don't just eat glucose. We eat all sorts of things. We eat other sugars, fructose, sucrose. We eat fats, we eat proteins. And so we have to break all those things down and oxidize them. So these pathways that we talked about, one reason why we learn about them is because they are really central and core pathways. So you can see that we can break down sugars that way. If we have fatty acids, we can break those down. Fats, we can break them down into glycerol and fatty acids. You can see they enter at different stages, the glyceraldehyde 3-phosphate stage, the acetyl-CoA stage, and that all feeds into the citric acid cycle. Proteins, we have 20 different amino acids. And these different amino acids, they feed in at different stages of this, this whole thing. So we can use these pathways to break down other fuels. The other thing to notice is I put these dotted lines in here, right? This is not in the textbook. I put these dotted lines in, is that a lot of these enzymes can be run backwards. So you probably know if you eat too much glucose, you eat too much sugar, what happens? You're going to turn it into fat. So how does that work? By running these pathways backwards. We get sugars, we make acetyl-CoA, and eventually we make fatty acids. You can also consume food and make some of your own amino acids, and that's as a result of running some of these pathways backwards. It does involve other enzymes, of course, not just the core ones that we learned, but you know, just know that these pathways are all intertwined, and that's kind of a good point. So just looking at the time, I want to kind of talk about the next topic for just a minute or two, and probably let you off a minute or two early on Friday. Then I'm going to play this video for you. So I'll come back to that in a moment. Where's my, I'm missing, okay, well, let's do the Kahoot. And I'll see if I can find, in the meantime, see if I can find the diagram that I was looking for. If not, we'll just have to save photosynthesis for next day. So load up the Kahoot, and if we have time, I'll play that video for you as well. Here we go. Loading up the pin. So it's going to ask you some questions about respiration and photosynthesis. Okay, I will give you another 10 seconds to join, and then we're going to get started here. All right, here goes. Cellular respiration. 10 questions. Glycolysis requires oxygen. Correct answer is false. Good job. So remember, the oxygen is only required of the electron transport chain. Number 2, where does glycolysis occur? Missing a word in that sentence. It occurs in the cytoplasm. So glycolysis is found in all organisms, even if they don't have a mitochondria. So it occurs in the cytoplasm. Question 3, the oxygen that is needed for cellular respiration is needed for which process? I just told you that. Hopefully you remember what I said. And the correct answer is the electron transport chain, and nobody got that one right. Pyruvate oxidation is where we break down pyruvate to acetyl-CoA. There's no oxygen involved there, although we do release carbon dioxide. All right, no change in points. Question 4, ATP is one of the products of glycolysis. What are the other 2 products of glycolysis? NADH and pyruvate, so good job. Okay, question 5. In the absence of oxygen, yeast cells undergo fermentation, which yields what? So this is going to tell me who did the lab yesterday, right? Or who just listened 5 minutes ago. I won't hold it against you if you don't get the right answer. So the correct answer is CO2 and ethanol. And of course, ATP. Question 6. During stage 2 of cellular respiration, acetyl-CoA enters the what to form ATP? So another way to think about this is what chemical pathway starts off with acetyl-CoA. I borrowed this Kahoot from someone else. Sorry, I meant to reword some of these questions that are a little wordy. So the correct answer is the citric acid cycle. So good job. Okay, question 7. Stakes are getting higher. Which step of cellular respiration generates the most ATP? Again, I don't like how this one was written because I don't like the right answer. The right answer is apparently the electron transport chain, and that is actually not correct. Remember, the electron transport chain is just half of oxidative phosphorylation. The ATP is not made there. The ATP is made in the final step, which is ATP synthesis. So sorry, I borrowed this one from somebody else. I guess I didn't read it very carefully. But good job if you got the right answer. Electron transport chain was the most correct answer there. Question 8. Which types of cells can undergo fermentation? So the correct answer is both. Remember, we had lactic acid fermentation and we had ethanol fermentation, and those are found in different cell types. So both is the correct answer. All right, 1 or 2 more questions. Let's see how the score plays out. The production of yogurts and some cheeses are the result of what process? So I guess I mentioned this in passing. So to get this one, extra bonus points. Oh, wow, good job. Everybody got that one right. So I'll have to put this one on the midterm for everyone who's present. And you don't need to share with the people who are not present, but it's up to you. Okay, last question. Which process is the most efficient at generating ATP molecules? I think we kind of, no, okay, a little bit different from the other questions. Sorry if I didn't address this one directly, but definitely aerobic respiration because you're making the most ATP molecules there. Okay, let's see how the score goes, and looks like we're almost out of time here, so I'm just going to finish up here in a moment. So good job, Tristan and Nina and Levi. Okay, good job, folks. Okay, so sorry, I had meant to show you this video. I'm not going to do that right now, obviously, but you can review it on your own time if you download the PowerPoints. It's a good review of the whole process, well animated, and hopefully you get something out of it.